Identify the dorsal and ventral surfaces of the squid. The dorsal side is dark. The ventral side is light and bears the siphon, a funnel-shaped structure that extends between the eyes. You might ask students how the body of the squid is oriented in the water column as it swims, based on this counter-shaded pattern. Examine the structures that are derived from the molluscan foot. There are 8 arms, 2 tentacles, the head, and eyes. Notice the siphon. The mantle is the largest part of the body, with prominent fins. Notice that the octopus's mantle is bulbous and lacks fins and a stiffening pen. Discuss how these features are suited for the benthic lifestyle of this cephalopod. On the dorsal side of the animal, find the point of the pen. Rubbing the squid's skin with your finger sometimes causes the skin to darken by spreading out the pigments within the chromatophores. Skin removed for microscopic examination should be laid out flat on the slide. Notice that suckers are present along the entire length of the arms, while suckers are only found on the distal end of the tentacles, in an area called the club. To expose the buccal bulb, cut along the midline. Appendages are numbered 1 through 5 on each side, beginning from the midline of the dorsal side. So in this ventral view, the scissors are cutting between the 5th arms of the right and left side. You might feel a crunchy structure with your scissors. This is the cartilaginous cranium. Food travels in a posterior direction through the esophagus into the digestive organs within the mantle cavity. Removing the beaks is simple, but sometimes it is difficult to find the radula. Look between the beaks and in the buccal bulb for a structure that looks like a small strip of plastic. Under the microscope, the radula has a toothed, rasp-like appearance. Don't be discouraged. It may take several squid before you learn to recognize and find this particular structure. Remember to keep your scissors pointed up while you make the initial cut to avoid damaging the internal organs. After opening the mantle cavity, place the specimen in a shallow container and cover the specimen in water so that the delicate internal organs can float, making them easier to see. Loligo is an annual species that dies after spawning, and most loligo are caught during spawning season when most of their energy is allocated to gonad development. Therefore, the liver is usually degenerated and small, as seen here. Lateral to the liver are the siphon retractor muscles, which control the direction of the siphon. The stomach is found posterior to the right gill. Since loligos cease feeding during spawning, their stomachs are usually empty at time of capture. The intestine is a clear tube running the length of the ink sac. The orange-colored kidneys excrete nitrogenous wastes into the intestine, and these wastes leave the body through the anus. The 3 hearts are light-colored bags of thin tissue. You may want to cut the gill connections to the mantle. This makes it easier to see the underlying structures. The gill hearts pump the colorless blood into the gills. Then the oxygenated blood enters the systemic heart to be pumped to the rest of the body. Discuss how this system compares to the atria and ventricles of the mammalian heart. The following are several views of the systemic and gill hearts. Next to the mantle retractor muscles are the stellate ganglia. Look along the sides of the mantle to find the largest nerve cells in the animal kingdom, the giant axons of the squid. You may wish to have students examine a section of an axon under the compound microscope. The reproductive organs take up most of the space within the mantle cavity. Females are distinguished by their prominent nidamental glands. The size and shape of the gonads may vary, especially if the animal spawned just before it was caught. The nidamental glands need to be removed to see the other female reproductive organs. Cut carefully at the thin mesenteries connecting the glands to their underlying structures. The nidamental glands secrete the outer capsules of the egg masses. The oviductal gland secretes the individual egg cases. The male reproductive system has three main parts. The single testis is located in the most posterior region of the mantle cavity. The spermatophoral gland is usually the largest structure and bears the vas deferens, leading to the coiled spermatophoral sac. The penis does not enter the female's body. The male's modified hectocotylus arm collects spermatophores from the tip of the penis, then transfers them into the female's mantle cavity during mating. The hectocotylus is the fifth arm on the right side, as seen from the dorsal view. In this ventral view on screen, the hectocotylus is the first arm off the midline on the left side. The suckers on the tip of the hectocotylus are tiny and comb-like, unlike the rounded suckers on all the other arms. Spermatophores are easily viewed by the naked eye, especially if the spermatophoric gland has torn. They are interesting to view under the compound microscope. Sperm cells exit when the spermatophore ruptures. Removing the lens is a simple process. Notice the great amount of dark pigment. What could be the adaptive value of this pigment? The last step is to eviscerate your squid and remove the pen, a relatively simple procedure.